Friend, welcome to Biology Synthesis with World Affairs. Today we will discuss about GPCR. GPCR GPCR is an signaling molecule. It is the largest family of cell surface receptors, and they mediate most response to signal from the external world as well as signal from other cells, including hormones, neurotransmitter and local mediators. Our sense of sight, smell and taste depend on them. There are more than 800 GPCR in human, and in mice there are about thousands concerned with the sense of smell alone. Today here we will see a ideal GPCR protein structure. As you can see that it is a 7 transmembrane protein. These are just go around the plasma membrane, and it is an one terminal and here is an other terminal. By this terminal they interact with the ligand, and by this here also 3 heterotrimeric protein alpha, beta and gamma present. This alpha have GIF activity and beta gamma also act on other target protein. So in next slide we will see that how this alpha molecule act on other surface. Here, as I told in previous slide, that this is the alpha molecule and this is an beta gamma complex. This alpha molecule has an point RAS domain and also have H domain where GDP is bound. Here we can also see this is the GDP binding site of alpha molecule. This is an beta gamma complex. Okay, next, as I said that this alpha molecule have, when it will be inactive it will bind with GDP, and when it will be active it will have to bind with GTP. So the process of conversion from GDP to GTP is mediated by gap. A alpha molecule itself has an gap function and also have gap function. So by the gap activity the GDP will convert it into GTP. After the work the GTP again will be converted into GDP by gap activity. GTP is activating protein, this means, and gap means guanine exchange factor. Next slide we will see. Friend, so we can easily show that here an signaling molecule bind denoted by red, and the GPCR activate. After that here an alpha and beta gamma complex separation will be occurred, and after that this alpha molecule will goes to the adenylate cyclase that is present on the plasma membrane. This adenylate cyclase will bind with alpha complex and it will convert the ATP into cyclic AMP. This cyclic AMP also will convert it the inactive pKa. pK means protein kinase present in cytosol. This will also convert pKa into activated pK in the cytosol. This activated pKa then pass from the cytosol to the nucleoplasm by nuclear pore complex, where it will bind with the inactive CRAVE. This inactive CRAVE will result in the formation of active CRAVE. This is the CRAVE binding site. Also we can know that in CRAVE there are two subunit. One is regulatory subunit and other is catalytic subunit. The catalytic subunit will then go under gene transcription and stimulates the gene transcription process in various places of DNA. Thank you, friends.